First chapter of your textbook. In this chapter, we're going to explore the invisible world as well as, the whole course is about the invisible world, microbiology. World organisms that we can't see with what they say is the naked eye, so without a microscope or tools like that. Microorganisms are everywhere. They're ubiquitous. I'm talking from volcanoes to really salty waters, inside our guts, on our teeth at the end of the day in something called a biofilm. They are just everywhere. Now, most microorganisms don't cause us any harm. They don't do anything for or against us. There are definitely some that cause harm, and there's plenty that actually help us. So we're going to dive into all that within this whole course. Um, and this is going to help us understand disease, medicine, the world around us, the world inside of us, and life itself. So it does. They influence our health, our food, our environment, and even at a global scale, the global ecosystem. Can you imagine a time where people didn't know that there were tiny little organisms that they couldn't see that were intensely impacting their lives? Just imagine that moment when people started to discover there were things all around them, ubiquitous, right? Meaning all over, um, that were causing disease, or they were the reason that they could make certain kinds of food, right? Fermenting things. Um, and then as they started to learn about them, they could utilize those microorganisms to do even more. But it must have been a pretty significant moment in time when humans started to associate things you can't see with things that are happening around you that you can see. So, yeah, we're going to look into all of this, and I want you to be able to describe historical events associated with the birth of microbiology. So, who were some of those big players in the beginning? Describe how these causes of sickness and disease were explained in ancient times prior to them figuring this stuff out. And then, in addition, so how did our ancestors improve food and use invisible microbes? So, pasteurization, all that good stuff we're going to talk about. Humans have been using microbes for thousands of years. As early as 7000 BC, people were fermenting beverages and bread using yeast, specifically Saccharomyces cerevisiae. But even though they had no idea that yeast was actually alive, was a single-celled organism, they understood that fermentation improved the flavor, improved preservation, and it improved nutrition. And maybe they had an idea that it actually improved safety as well. This is one of the earliest examples of biotechnology. So using life, bio means life, and technology, so technology to benefit humans. So humans have been using microbes for thousands of years. As early as 7000 BC, people were fermenting beverages and bread using yeast, specifically Saccharomyces cerevisiae. Even though they had no idea that yeast was a living entity, these microscopic single-cell organisms, they didn't know they were alive, but they understood that fermentation improved flavor and preservation and nutrition, and they likely understood that it improved safety. They would often drink fermented drinks like beer, because the boiling process and the fermenting process actually killed a lot of the pathogens. Now, they had no idea about pathogens, but they probably likely could see that you didn't get as sick if you drank, maybe it was a low alcohol beer more regularly than just the local water. So this is one of the earliest examples of biotechnology using living organisms to benefit humans. So using technology, altering life, and it's benefiting humans. Patsy the Iceman gives us fascinating evidence of early interactions with microbes. So scientists found that he was infected with parasites, infected with bacteria, but he also had carried with him a fungus that we now know as an antibiotic, well-known antibiotic, and it also had laxative properties. So basically, he was carrying around medicine without really knowing that in the future that's what it would be called. This suggests that even in prehistoric times, humans may have experimented with natural treatments, even if they didn't understand the cause of the disease or the reason for the cures. I'm sure it was a lot of testing out things that led to some pretty bad side effects as well. But in this case, we can see that he was carrying something that actually may have helped him. Ancient civilizations tried to explain disease without knowing about microbes. They didn't even know they existed. But they believed that illness came from bad air or mal area. That's an Italian phrase which means bad air, and it probably reminds you of one of the many diseases out there, malaria. At the same time, people noticed disease could spread, which led to early quarantine practices. Engineering projects like the Roman sewers and aqueducts also suggest that there was an early understanding that sanitation affected health long before microbes were discovered. There were several ancient thinkers that made important contributions to medical thinking. Hippocrates argued that disease had natural causes instead of supernatural ones. At the time, many people thought that if you got a disease or a sickness or if you died, that perhaps you were being punished by the gods or something beyond people. And the same with healing. They often associated, good things that were happening to being blessed and being healed by gods beyond and nothing to do with science or nature. So Thucydides carefully recorded symptoms during plagues, and Marcus Terentius Varro suggested that tiny invisible creatures could cause disease. So this was so much earlier than the microscope was invented. It's not anything he ever saw under a microscope. He was just guessing. He was observing the world around him. And you'll notice that a lot with scientists. It's a lot about observation, that first step in the scientific method. Sometimes I worry we're not putting down our phones long enough to kind of observe the world around us. But he observed that if you build your home, your little hut, or your abode near a swamp, that you would be more likely to possibly get sick. So he was warning people about not building near swamps. But that is kind of the beginnings of the germ theory. The modern germ theory now is kind of rooted in this. Microbiology truly began with Antony van Leeuwenhoek, who used powerful lenses to observe microorganisms for the first time. So he worked with threads, and he wanted to look more closely at them at one point. He invented something that would magnify items at 300 times. Now, the microscopes we use in the lab, a high school, college lab, those light microscopes, we typically look at things under 400 times. So he was doing pretty well as far as inventing that. Robert Hooke, who looked at plant cells, he didn't have as fine of lenses, and he didn't get to see anything that was actually alive under his lenses. So Antony van Leeuwenhoek, he was so curious. I'm going to attach a video, which is a really good video. It's a very nice overview of his contributions. But I have read in other places that he did things like when he was sick, he would look at his vomit under the microscope and his diarrhea, and he would look at semen. Anything he could get his hands on, he wanted to look at under the microscope. Algae, things like that. So very curious, again, a curious person. So imagine being that first person seeing these invisible things finally visible, alive, right in front of you. How do you explain that to anybody? They might think you're a little crazy. Um, but what an exciting moment, I'm sure. And I'm sure he just couldn't satiate the curiosity by looking, kept looking at everything under the microscope. Um, he was Dutch, and yeah, there's just so much about him. I think it's a really cool part of the story. So later, during the golden age of microbiology, when lots of things were discovered, scientists like Louis Pasteur showed that microbes cause fermentation. So they'd been fermenting foods for a long, long time and beverages, but he actually made that connection between the microbes causing the fermentation and microbes causing disease, right? We had some earlier um, curiosity seekers, scientists thinking about that as far as there could be little things you don't really see that could be causing disease, but he was really the one that put that together, kind of solidified that idea. And then you have Robert Koch, proved specific microbes cause specific diseases. So he connected, one disease is going to be caused by a specific type of organism. Now we know that pretty well, right? We all lived through that COVID pandemic. And well, unless this video is listened to a long time from now, but we know, there's a specific microbe that causes that. There's a specific microbe that causes flu and strep throat, right? But people didn't know that back then. These are all connections made during this time to lay a foundation for the germ theory. So these discoveries changed medicine forever. Now that scientists could observe microorganisms, the next challenge was organizing and classifying them because, as you know, there are a lot. In this section, we're going to explore how microbes are grouped into species, how classification systems have evolved over time, and how modern science uses genetics to organize life. Carl Linnaeus developed a system for naming and organizing living organisms called taxonomy. So just like all the various disciplines have a systematic way to organize things, like mechanics, you're not going to have all your tools in one bucket. Chef, you have your cold stuff, your stove, your pots and pans, your spices, and they're not all in one place. And all of these different disciplines also have their own language and terms so they can communicate with one another. So in this world of science, we have this taxonomy where there are grouped organisms based on shared traits. And he also introduced a standardized way of naming them. While his system focused on plants and animals, it laid the foundation for later microbial classification. So first we're starting with plants and animals, etc. So there's this wonderful way he contributed to the field of science. The more that scientists discovered, it got more difficult to keep track of everything and to organize this. So that's the bright side, and there's a little bit of a dark side to Linnaeus. And I did include a video per that. Maybe you're interested, maybe you're not. But it kind of delves into what happens when one person tries to classify humans into different categories. So, as you might guess, that's going to be problematic because if you have one worldview or you haven't really encountered people that aren't like you, there's going to be some stereotypes that you use when classifying. So, two things can be true at the same time. You can be a person that has contributed something amazing to the world, and there's this other side to a lot of the people in our history, which I think we need to give a nod to, so we don't repeat history, at least not the parts that hurt other people. So that's my 2 cents, and enjoy my videos. As scientists learned more, the classification systems evolved. And then Ernst Haeckel came along and he proposed new kingdoms like Protista and Monera so they could include the microorganisms in this classification system. So I did a little bit of a deep dive on Ernst Haeckel. I didn't know a whole bunch about him, so now you're going to know more about him because I learned it. It was really kind of difficult to find videos that I felt were fair, I guess you would say. A lot of them had a very religious slant on trying to like debunk his ideas. So here's what I came up with my deep dive. So he was a 19th century German biologist who strongly supported evolution after Charles Darwin. So he supported those ideas. He's best known for promoting the evolutionary theory in Europe. So he spread the word there and coining the term ecology. So, um, and then he's famous for his beautiful drawings. You have to watch the video that I attached. It's just, they're just incredible pieces of art. Different jellyfish and, whatnot. But there's these other drawings he's famous for, but it's controversial. They're drawings of embryos. So, story goes that, he made a strong version of evolution. He didn't just support evolution, but he made this bold claim. Ontogeny recapitulates phylogeny. So that means the idea that he suggested embryos literally replay evolutionary history. So human embryos go through a fish stage. So it directly challenged creationist views, was later shown to be oversimplified and partly wrong. So then the critics, especially in religious debates, often use Haeckel as a gotcha example to discredit evolution more broadly. So the embryo drawings became that flashpoint. So he made drawings comparing embryos of different species that look very similar. And if you go look at, various organisms, their embryos, they really do look very similar. So they're partly, they do look similar if you do go and look, but embryos do not share similarities. They do share similarities in early development, but Haeckel exaggerated the similarities, he simplified the differences, and he reused similar shapes across species. So his drawings were not fully accurate, but the core idea that shared developmental features due to common ancestry is still supported. And you can see that for yourself when you go look these different embryos up, if you find a legit site to look at those. So overall, there is he didn't add into the evolution foundation of science very much. But he didn't completely fake everything, as you'll, find people are accusing him of, but he absolutely overstated a real pattern. So, yeah, and that's the rabbit hole I went down, but it was very interesting. And I don't know if I already mentioned, there was also this racist side to some of his work, just like Linnaeus. Just hierarchical ranking of human groups, people being more evolved than others, which is a real shame because none of that is true down to the DNA level, as we know. But it did not help that, as far as the way we treated each other throughout history. But like I said, the good and the bad of it. Take a look at his drawings. They're incredible. The art is incredible. Okay, I think that's it for that. Oh, Robert Whitaker. I have to talk about Robert Whitaker now. Almost forgot about him. So later he separated fungi into their own group. So these changes reflected growing knowledge about evolutionary relationships among organisms. So I don't know a whole bunch about Robert Whitaker as well. But I don't know if I want to go down that rabbit hole. Might be we might be here for another 10 minutes. Slide highlights how classification systems are not fixed. They change as new information becomes available. As technology improves, scientists continually reevaluate how organisms are related, refining that tree of life. So we also can refer to that tree of life as a phylogenetic tree. And all of the branches that are put on there. Maybe you've built a family tree before for an assignment when you were younger. These particular organisms, this is how we guess how things are related using morphology, so what the organism looks like, where it's located, fossil records, and now, of course, we can enter in DNA evidence to see how things are related. It's a tree of hypotheses, right? The best guesses we can make with the information we have and the technology we have. And as I said before, science isn't afraid of more information. Um, this is just the truth as we know it right now. So I talked a little bit about the morphology and the fossil records as far as how we can classify the relationship between organisms throughout time. But modern taxonomy relies heavily on genetics. So by comparing DNA and RNA among the different organisms, especially ribosomal RNA, scientists like Wois and Fox discovered that life can be divided into 3 domains. So we have bacteria, and we have eukaryote, right? Eukarya. And then Wois discovered archaea. So we have these 3 domains with everything else kind of fitting in within those phylogenetic trees. This genetic approach revolutionized how we understand biological relationships. So check out the little video I have of Wois. And Bill Nye, the science guy, he's narrating it. So I don't know if you know who that is. Some of you may have grown up with listening to him in school. Others of you, maybe it's before your time, but just thought I would mention that. Microorganisms are named using binomial nomenclature, which includes a genus and a species name, such as Staphylococcus aureus or Escherichia coli. Scientists also identify different strains within that species. And strains are really important when it comes to medicine, especially in disease. So a bacterial strain is a genetic variant, a subtype, or pure culture derived from a single cell within a bacterial species that exhibits unique phenotypic or genetic traits. So your phenotype, something you see, like a phenotype for you might be your brown eyes, or maybe you have a large liver, something that, but the genes behind that are the things you can't necessarily see, right? You get an allele from mom, you get an allele from dad, and maybe you have brown eyes, but maybe an allele from mom was actually blue eyes. And then a brown-eyed allele from dad. That's kind of simplifying that. But with this, you're looking at those genetic differences or phenotypic differences. For example, there are various Escherichia coli variants. So you could use something in the lab that's very safe to use and it's not going to cause a disease. It's not a pathogen. And then there's definitely E. coli, like O157H7, that's highly pathogenic or, makes you very sick. There are different probiotics. Let's see what else. Looking here. But medical microbiology, identifying specific strains helps diagnose disease, determine treatment, and identify antibiotic resistance. In biotechnology, you can utilize, you can engineer strains that are used to produce medicine, like insulin. So E. coli is used to create human insulin. So you have to alter E. coli to produce that insulin. So yeah, there's a lot of different ways that strains come about. But The overarching is the bacterial species, and then under that you have these subtypes or different strains. Beyond that, let's see. So Bergey's manual is a manual that was originally written in 1923, and the more and more bacteria that are identified are written about, and the manual is constantly being updated. But that's where you can go. It's kind of like it's been referred to as um, the Bible of classification for bacterium. So that's about it. Next, we'll survey the major groups of microorganisms. We'll compare bacteria, archaea, eukaryotic microbes, and viruses, and we'll begin to see how microbiology connects to many scientific fields. So take a look at these objectives and let's move on. All microorganisms are small enough to require magnification. There's one exception I know about, there's probably others as well, but the tardigrade, or better known as the water bear, it's actually considered a microanimal. It's multicellular. It, but since it doesn't get much bigger than 1 millimeter, that's technically not too small to see without a microscope, but to see it clearly, you need a microscope to study it and to look at it, close enough. Not many people can see something at 1 millimeter. But some of these microorganisms are unicellular, so its whole living being is just one cell. Some are multicellular, right? Meaning that it takes more than one cell to make up its whole its whole body or its whole entity. And then some, like viruses, are acellular. A means not, so not cellular. It doesn't. It has to get into a different cell to be able to exist. So microbes exist in all three domains of life and display incredible diversity within those domains. Bacteria are prokaryotic cells, meaning that they lack a nucleus. So that has ancient Greek roots. Pro meaning before or in front of, and karyon meaning nut or kernel, and that refers to the nucleus. So prokaryotic cell is before the nucleus evolved. We have eukaryotic cells, and we have nucleus or nuclei in our cells. So that is the root to that word, and it helps you remember that prokaryotes are the ones that don't have the membrane-bound nucleus. Bacteria, they can be found just about everywhere. Some are helpful, like probiotics or lactobacillus, which we use in the creation of yogurt. Um, and some help us digest our food, break things down. Some are harmful pathogens. They cause disease. And this isn't just to humans. These can be helpful, harmful, and harmless to other organisms as well, plants, fungi, etc. So you have harmful ones, and you're familiar with various diseases, Salmonella, Streptococcus aureus, right? And we have harmless ones, and sometimes they're called commensals. And that means they just kind of hang around and they don't cause any harm. Typically, they're benefiting without causing harm or not even really help. So we have various E. coli strains that are actually they're not pathogenic to us. Some E. coli are pathogenic, but some aren't. And those strains, they kind of just reside harmlessly in our guts. And they may assist with digestion sometimes, but typically they're just hanging out. They're benefiting. They love it in there. And beyond that, bacteria have various shapes and metabolism. So they have just a wide variety. You can see the pictures on the slide there, the various shapes, and that's going to dictate different movement and different portals of entry, the way it can get in to our cells. So they play a major role in health and environment, all these various kinds. The metabolic diversity, um, some of them are photosynthetic, meaning that like plants, they can photosynthesize. So there's cyanobacteria that can do that, and that's just, they're self-feeders in that way. And most of them, are not there's non-photosynthetic. So like I said, the DNA they don't have a membrane-bound nucleus, so the DNA is not kept in a nucleus like ours is. They're just kind of kept in a region, a nucleoid region. And then you have cell walls, and depending on the type of bacteria, the cell wall is going to be different. That will play a role into what kind of medicines can help kill them. So the antibacterials or antibiotics that we take, the type we take will depend on the wall that that bacteria has. And yeah, so we describe them. Sometimes it's nice and a little easy for us. The name of it has the shape right in it, like Staphylococcus, right? That's that cocci, or as you see in the picture, coccyx shape. We have, what's another one? Let's see, Streptococcus again. We have Bacillus, like I said before, Lactobacillus, so that'll be that bacillus shape. Yeah, there's a big variety of the shapes and metabolism. Archaea are also prokaryotic and unicellular, but they are genetically distinct from bacteria. Those two groups used to kind of be all put together in one group, but Carl Woese, we talked about him earlier. He's the one that discovered how different they were from bacteria. We're actually more related to archaea than we are bacteria. So they can live in extremely extreme environments, like you see in the picture here on your slide. That's likely from Yellowstone. Um, and that's an incredible place to go if you ever get a chance to go there and see the hot springs. All those colors, though, those are the archaea that's in that particular hot spring, that hot pool. Yeah, if you ever get a chance to go to, it's one of the most amazing places in the United States. So they can live in very salty environments, very cold environments. They are extremophiles. Phile means love. Extreme, they love the extremes. So far, this is good news. There are no archaea that are known to cause disease in humans. Whoop! So that's awesome. All right. But you got to go to Yellowstone. Extra credit. Protists are a diverse group of mostly single-celled eukaryotic organisms. So eukaryotic, they do have a nucleus that's membrane-bound where the DNA is kept. But they don't really fit into the other categories like fungi. They don't fit into animal. They don't fit into plant kingdoms. I was thinking of this as I was looking at this, thinking of it like a junk drawer in your house where these are things that you don't know where to put in your house, so you throw them all in the junk drawer. But they are mostly found in moist and aquatic environments. So they could be amoebas, they could be algae, they can photosynthesize, they can be non-photosynthetic, so meaning heterotrophic, needing to get their food from something else. Yeah, they're kind of all over the place, but they can be very useful in products like agar and biofuels. So agar is something that we actually use in the microbiology lab quite a bit. Protozoa are highly diverse. They might be free-living or parasitic, and several can cause human disease. So I guess we'll be looking into that later. Fungi are eukaryotic organisms that include yeast and molds. So again, they have eukaryotic cells like we have. If you've ever had a fungus infection and you had a hard time getting rid of it, well, that's because it's hard to kill fungus when their cells are very similar to ours, right? Bacterial cells are prokaryotic, so there's a big difference. So they have cell walls made of chitin. Other things that are made of chitin, like crab shells. So chitin is a natural biodegradable polysaccharide. So saccharide, you think sugars, you think carbs. Poly means several. So there's several sugars put together. So they are found not only, like I said, on fungal cell walls, but also crustaceans and insects, right? That hard exoskeleton. So, fungus also plays a big role in decomposition, so breaking things down because they need to feed off of other things. So we know that they're in our food production, whether it's part of the production or, part of the product itself, like on pizza. And in medicine, you are familiar with penicillin, right? So in antibiotic production is something that is associated with various types of fungi. Almonds are parasitic worms. While they're not microorganisms as adults, their microscopic eggs and larvae are studied in microbiology because of their role in disease transmission. Um, they get transmitted primarily through ingestion of eggs or larvae from contaminated food, water, or soil, and direct skin penetration sometimes. So there's soil transmitted helminths like hookworm and roundworm. If you're walking around barefoot, or if you were gardening and then you bite your nails, or I always worry about that. I'm a nail biter, so I always, I'm always washing my hands. Um, or possibly, your animals bring them in on their feet because they're walking around. But undercooked meat, undercooked fish, and fecal oral spread, insect bites, etc. So there's a lot of ways to get these parasites. So wash your hands. Viruses are acellular. A means not, so they don't have a cell. They're infectious agents made of genetic material and protein. So they have to infect a host to um, to kind of come to life. They're not considered alive. So if there's a flu virus right there on your counter, it's not like it can move or crawl to you. That's why the masks were very effective during COVID because, the viruses, they're tiny. So if they could walk or move, I suppose they could get through your mask, but they travel in droplets like sneezes and talking, and those droplets are too big to get through those masks. So they're not considered alive, they're acellular, they must infect a host to replicate, to make more of themselves, and they can infect all forms of life, including well-known examples like coronavirus, like Ebola. If you look there on your slide, you can see the bacteriophage that actually infects bacteria. So it's a virus that attacks bacteria. So, yeah, the viruses are varying degrees of virulence, meaning, some aren't as harmful as others. As I guess we kind of know that, right? Because something like Ebola virus, very deadly. Something like a cold virus, not quite as deadly. Lots of variation. And finally, microbiology is a broad field that includes many specialties such as bacteriology, virology, immunology, and mycology, and so on and so on. Together, these fields contribute to medicine, biotechnology, and everyday life.